Hello students, welcome to week number 5 of Biology 120 Lab. It's our DNA week and next week is genetics, so that's going to be exciting. So let's take a look at what we have going on. Last week we looked at cellular respiration with those peas, and hopefully those germinated peas actually did some cellular respiration and took in the gas, took the oxygen out of the gas in order to do aerobic respiration. I hope you saw that in your experiment, but I'll see when you turn in your lab reports. So cellular respiration, remember, is a way for organisms to break down food, to make energy in the form of ATP, and they use oxygen when it's aerobic respiration. But there are other forms, anaerobic respiration and fermentation, that do not use oxygen, and those are types of cellular respiration also. Okay, this week is all about DNA. DNA, one of my favorite molecules. DNA, I don't have it very straight, but anyway. DNA, deoxyribonucleic acid, the molecule of heredity, and how traits are passed along into the next generations. So we are going to take a look at that this week. I'm starting off here with milestone 2 because it will be one of your assignments this week. And this milestone is your experimental design. So you're going to tell me how you're going to do this experiment. Now, I realize that some of these are online, like the Manduka caterpillar lab. It has already been performed for you. There are several different temperatures that you can look at. Okay, some of the others, like the ice or the nervous, you'll actually be doing something, so you'll have to explain that, what you have chosen to actually do. And for the air pollution one, you're going to be looking up data. So you're going to describe to me what you're going to be looking for, where you're going to look, what particular pollutants are you looking for, over what time period, in what location, all that sort of thing. Okay, so first of all, I have graded all the milestone 1 assignments, so make sure you go back and fix those, because you're going to have to fix that in order to put that into the final project. So fix milestone 1, look at my comments in the graded rubric and in the feedback section, because I leave comments in both of those sections. Make sure you use the correct format. Always look at that on your guidelines and rubrics so that you do things the correct way, okay? And include all the steps to your experiment. Step 1, I'm going to do this. Step 2, I'm going to do that. You get it. So write down all the steps for your experiment. All right, so the 2 labs that you have this week, one is the cheek cell DNA extraction and the other one is a PCR simulation, which I'll explain in a second. So lab number 5, the cheek cell DNA extraction. This is a simple little experiment where you are going to extract your own DNA. You're going to chew on the inside of your mouth while having a little bit of sports drink in your mouth. And just that mechanical action will brush off some of your cheek cells, and you'll be able to collect those in your sports drink after you spit it into your cup. And then you're going to do a couple of things to it to break open the cells and then to isolate the DNA. I have pictures here of what mine look like, and you can see the cloudy, stringy stuff there. That is the DNA. So you're going to be able to see your own DNA. This is what they do if you're arrested for a terrible crime and they collect your DNA. But it's also the way that I did Ancestry.com, which was an interesting experiment because I actually found a half-brother that I did not know that I had. So that was an interesting thing for me to do. But I did it the same way as I did this experiment, basically. They just tested my DNA after I sent it off. So a couple of things about this lab. If you see CC written on a cup or a syringe or a pipette or anything, a lot of the time in medical practices, in medicine, you see cubic centimeters, cc. Okay, a cubic centimeter is the same thing as a milliliter, all right? They're exchangeable terms because they are exchangeable volumes, just so you know that, okay? You're going to be using ethanol, which is provided in your kit, but I need to caution you, it has to be extremely cold for it to be able to extract DNA out of the solution that you have. Extremely cold. So you need to put the ethanol in your freezer at least overnight. I know in the lab it tells you that you can put it into a bucket of ice or something like that. That is not going to be cold enough. You need to put it in the freezer overnight and take it out right before you need it, because it has to be ice cold in order for this to work. So I'm just warning you about that. Otherwise, you're going to be doing it again. This is a quick lab. It does not take long for this whole process. Okay, so that's a good thing. And you're going to take pictures like I took pictures right here. You're also going to do a PCR simulation lab. So what is PCR? PCR stands for the polymerase chain reaction. And all that means is a polymerase is a molecule that makes a polymer. And that only means a whole bunch of subunits stuck together of something. Well, that's what DNA is. A polymer. It's got all these little subunits stuck together to make the molecule DNA. Those little subunits are called nucleotides. So when you put a whole bunch of nucleotides together, you get either a DNA or an RNA molecule. So what is PCR? Making a whole bunch of copies of a polymer. That's all it is. Making a bunch of copies. And we want to make copies of DNA. Now this is a very simple process. It's just we had to figure out the right components and the right temperatures for this to actually work, where we could amplify or copy certain sections of DNA over and over and over again until we had enough copies where then we could take a look at that and figure out what the DNA sequence was, or the RNA sequence also. The sequence or what kind of information is contained there. That's the purpose of PCR. Back in December, when I got sick with a respiratory infection, I was afraid that I had COVID, of course, even though I've been vaccinated and boosted. But I went to my local Walgreens, made an appointment, went to Walgreens, and I asked them for the PCR test for COVID. It is the most sensitive one because if you have a single copy of the RNA from SARS-CoV-2, then that PCR test can pick it up. The other tests are not as sensitive as the PCR test, but PCR takes a little bit longer. It usually takes at least a day to run it, for the lab to actually run it. And of course, you know, it takes time to get the sample there and then for them to report and send the report somewhere. So anyway, PCR takes a little bit longer than some of those other simple tests that you can take. But it has been very widely used during this time of COVID. PCR. So that's a great example of why you need to understand it, because we have been using it for these last 2 years. So answer the questions. We've got a PCR simulation video that you go and watch, and then you'll understand how it works, and you're going to answer some questions. So you got 2 lab reports to turn in, one on the cheek cell DNA extraction and one on the PCR simulation. And then you've also got a milestone 2 to turn in, which is how you're going to do your experiment. Okay, a word about next week. So next week is the genetics lab. And you are going to be growing 2 sets of seeds because you don't have enough petri dishes to just run this one time. So you have to grow seeds in 2 different sets next week. Okay, you're growing something. So it takes days for the seeds to germinate and the little plants to germinate, right? This is going to take at least the whole week for this growth process. And then you have to figure out what all that means, and you have to do Punnett squares and all that, which we'll get into next week. But I'm just warning you ahead of time that it's going to take the entire week for you to grow, germinate basically, these seeds. If it were me, I would start this coming Saturday. I would start my seeds. It doesn't take much time to do any of the setup or check on them or any of that, but it does take time for them to germinate. So if it were me, I would start like at least this Saturday, even though we're still in week 5, and start those seeds to make sure everything goes okay and that you get results. If you grow them for a shorter period of time, you will not get the results. You can't tell the difference between them, and you have to in order to do your counts. So please, please realize that this genetics lab is going to take a solid week of growth because you got 2 sets of seeds. So start early, as early as you can. All right. And as always, if you need anything, if you have any questions, if you're coming up against some problems and you don't know how to do something, please let me know. Just email me at m.sigmon@snhu.edu. Put it in the general discussion board. I will be glad to answer questions there. Anywhere you want to get in touch with me, you can. If you need to do a little video call this week because you're having trouble, just let me know. I'm glad to set one of those up for you. Okay, I hope that you have a wonderful week this week with DNA. DNA. All right.